Welcome to the PDBe webinar. In this webinar we'll cover the following topics. Who are we? Searching the PDB and the PDB entry pages, both at our website pdbe.org. The PDB is an archive of experimentally determined three-dimensional structures. These include protein, nucleic acids, and complexes of both protein and nucleic acids. These can vary from very small molecules, such as this copper scavenger shown here, to massive cold viruses shown here. Since the inception in the 70s, the PDB has grown steadily, passing the 100,000 entry milestone in May 2014. The PDB itself is run by 4 different organisations under the umbrella organisation the WWPDB. These are BMRB, which handles exclusively NMR data, PDBJ, and RCSB, and us, PDB in Europe. Together we produce the FTP PDB archive, which is available from each site and is identical from each site. In 2002, here at PDBe, we formed the Electron Microscopy Data Bank, which has been jointly operated between us and RCSB since 2007 and has undergone considerable growth during that period. Our newly designed homepage, pdbe.org, allows you to simply access the PDB using our single search box, which is shown in red in this image, and then we have other services available such as deposition, shown in the green box, and other PDBe services shown in the blue box. Our popular services are shown on the right of our web page, as are the latest statistics. Popular services include EM Search, PDB Fold, PDB PISA, our new REST API, sequence searching, and the new Empire archive for EM images. The latest statistics also provide a way in to find the latest PDB entries that are available and latest EM entries that are available. Searching the PDB using the website pdbe.org. We've redesigned our search from the ground up to enable you to use the following features. Auto-suggestion, so we will suggest results based on what you've typed in. Filters to help you narrow down the search results. Different views to help you analyse the results. Validation information presented directly on the research results page. Easy methods of changing the search and saving the search. I'll highlight these features in a bit. Before we get to that, we have to prepare the PDB data for searching. Because the archive is over 40 years old, some of the data is no longer in the cleanest format it could be. So we standardise the protein names from UniProt, so you can find your protein based on the name in UniProt, and standardise some of the terms in the PDB. This enables you to find, for example, all X-ray entries without having to worry about the spelling of X-ray. This data is available via pdbe.org through our webpage or through our API, pdbe.org/API. We also provide up-to-date sequence information and 3D domain information through using our SIFTs projects. The sequence information consists of up-to-date mapping between UniProt and PDB. This includes handling of obsolete UniProts. Also, we include the domain annotation of PFAM, Go and Interpro, and then the 3D domain information comes from Cath and SCOP. So to highlight the features that I mentioned using the search, I will go through 3 examples. So we have common user queries that we get in here. For example, how many species do we have structures of phosphoglucose isomerase from in the PDB, and what ligands they bind to? Starting from our home page, we only have one simple search box for you to type into. As you start typing, our auto suggestion comes into play, where it will narrow down the results and present you in categories all the different things that match what you're typing. In this case, we're interested in the molecule name, which will be phosphoglucose isomerase, of which there are 65 entries in the PDB. Once we select this, we get presented with our search pages like this, with the entries shown in the main pane, and then we have various features around the entries which I will highlight. At the top we have our search terms, and then just below that we have the different views that we have on these entries shown in the blue box, and on the left hand side in the red box we have the ability to refine our search, to add further terms to narrow down what we're looking for. In the main section we present an image shown in the blue box here. This is the image of the assembly of the entry, not the deposited coordinates, but the assembly, so you can see straight away whether things are dimers, tetramers, full viruses. You don't just have to rely on what's just in the entry. Just above this in the yellow box we have the validation sliders. This is directly from the new validation reports and is presented directly on the search results, so you can see which structure is the best. We enable you to sort these results, shown here, where we can sort by release date, quality, resolution, or PDB code should we want to. So to answer our query, how many species does phosphoglucose isomerase come from, we need to refine our search, and about halfway down we have the species name category, where we can see that there are 22 different species names here, so there are 22 different species from which phosphoglucose isomerase comes from, and we can select one of these categories to look at entries from just that species. So here I've selected mus musculus, and this then gets added to our search terms at the top of the page. Say we're now interested in which ligands are bound. Another one of these refined boxes shown on the left in green here is interacting compounds. So you can see that there are 10 different interacting compounds, and the number on the right hand side shows how many entries these compounds appear in. We can view this a different way. This is where these views come into it. So the compounds view that we have here enables us to see a different take on the PDB results, takes us away from the entry specific view. Here we can see that the results are sorted by compound, where the ligand is highlighted in the assembly images on the right hand side, and then a 2D representation of the ligand is shown, so you can easily see what the ligand is. When a ligand is found in more than one entry, the best entry is shown in the main entry, and the remaining entries are shown below this with their validation sliders and prints icons to depict the species that they come from, the method that was used to determine their structure. Another view we can use here is the protein families view, which allows us very quickly to see which PFAM domains are found in our results. In this case we can see that there are 4 different PFAM domains for phosphoglucose isomerase, and then the images highlight where the PFAM domain is within the structure. The next question we have to answer is, how many different human proteins are there in the PDB? So we start again with our simple search box, type in human, we select Homo sapien human in this case, we can see that there are 28,301 entries. We then need to narrow down our results to only show those which include protein, and this is shown in the red box on the left hand side. And then our macromolecules filter on the left hand side shows that there are over 100. It doesn't tell us the exact number, but it does tell us over 100. However, we can use the different views again to show the individual macromolecules that come from human, and this is in the blue box here. If we click on this, we get presented with a macromolecule view, of which we can see that there are 6,996 human proteins, distinct human proteins in the PDBe. And in each case, the individual molecule that is shown is highlighted in the assembly. So in the entry that's shown here, only one of these proteins is the one that we're interested in, and it's highlighted in blue. How would we find structures of LAC-I bound to DNA? So we do a simple search for LAC-I, and then we have a look down our search page to find assembly composition, of which we can choose the 13 structures that are DNA and a protein complex, and this gets added to our search results at the top, and we can see there are 13 different entries. We can simply change our search. Adding to it was very easy, but we can remove terms as well. So for example, we can remove LAC-I from this search, and this will leave us with all protein DNA complexes within the PDBe. Another feature we have here is the easy saving of a search. For example, here I've done a search for LAC-I, which is involved in a protein DNA complex. It's from Escherichia coli, and it was solved by X-ray diffraction. If I click Save Search, I get presented with a little pop-up. It now enables me to name my search, and then whenever I visit the search results page, I can see I have a 1 in my basket. If I click on here, I can retrieve this search and view it again. This is stored if you use the same browser on the same computer. This is stored on your computer without you having to log in. So to summarize, our new search includes auto-suggestion, filters to help you narrow down the search results, different views to help you analyze the results, validation information presented directly on the search results, easy changing, adding and removing of search terms, and simple saving of search without having to log in. PDBe entry pages at PDBe.org. The entry pages have undergone a significant redesign to help us answer common user queries. They've been separated into 5 sections. This is citation, function and biology, ligands and environment, structure analysis, and experiments and validation. And I will demonstrate how these sections are used to answer common queries. If we view an entry page, for example 1MQT, available at pdbe.org/1MQT, we can see the first section, which is shown in red, is the citation and entry information section, including the images. We have function and biology, which includes all Go term annotation, domains that are present in the structure, and if it's an enzyme, the reaction that is catalyzed. The structure analysis section, which gives you details about every single macromolecule in the entry and the assembly composition. The ligand and environment sections details the bound ligands and modified residues. And experiments and validation section gives details of the experiment and the full validation slider. So our common user queries that we have are: who has cited PDBe entry 2ASD? So if we click on the primary publication up at the top, we get presented with this citation page where we give you the abstract of the paper, the authors of the paper, which related entries this publication is found in, and then at the bottom highlighted we can tell you who has cited this structure. This is split into several sections. Starting at the bottom, we have articles citing this publication and reviews citing this publication, and then at the top we have reviews which mention this PDBe entry but do not include this publication in the citation list. This is available via our partners Europe PMC when the paper is open access and they're able to do free text searching of all open access papers to look for PDBe entries. As this paper is open access, we also have access to the images within the paper, which we present in a lightbox, so you can see whether you need to go away and read the paper or not before leaving the PDBe entry that you were looking at. The next question we have is: where is my protein in the assembly of a virus? So looking at the standard image gallery we have here highlighted in red, this shows the deposited coordinates with every chain coloured a different colour. And if we click on this, we get presented with a light box with a lot of the images produced for this entry. The first image we have is the same image that we saw on the home page, but then further on we have images which highlight each unique entity, so that's macromolecule or small molecule ligand within the entry, to show exactly where they are within the assembly. And then we combine these together and then colour each unique entity a different colour and show that within the assembly, so we can see at a glance where all the green proteins are, where the yellow proteins are, where the red proteins are, if there were any, for example. The structure analysis pages help us further look at exactly where proteins are within the structure. This highlights how many distinct macromolecules there are within the entry, and the green details button. This page then shows the details of every assembly within the entry and the unique molecules within the assembly are coloured a different colour, so we can see at a glance where they are within each assembly. Further down this page we then highlight each unique macromolecule. Here's protein, RNA, so a polymer for example, and exactly where these macromolecules are within the assembly. Then we might want to find which sequence and structural domains are present in our PDBe entry. Again, we'll use PDBe entry 2ASD for this. If we click in the box highlighted, the red box highlighted on this page, we see the details for the individual macromolecule. This presents you with a page like this, where we have linked 1D, 2D and 3D interactive diagrams. So highlighting or clicking anything in the 1D diagram will show it on the 2D and the 3D diagrams. Here we have a PFAM domain highlighted in both the 2D, 1D and 3D images, so we can see exactly where that is within the entry. We can change the setting on the 2D diagram to show different things. For example, here we can show quality, and we can show where all the outliers are within an entry. For example, this particular lysine we have highlighted, and it is shown up on the 3D diagram on the right hand side. The final question we will try to answer was which residues bind to thalidomide in PDBe entry 4v2y. In this case we will go to the ligands and environment section, clicking on an individual ligand, where we will see a 2D diagram on the left hand side and an interactive 3D diagram on the right hand side, so we can rotate the structure and have a look where the ligand is within its binding site. And then up at the top here in the light box we have the drug bound within the assembly, so we can see at a glance where it is within the assembly. So to summarise, our entry pages have been redesigned to answer common questions. They've been separated into 5 sections, which are citation, function and biology, ligands and environment, structure analysis, and experiment and validation. And thank you for listening, and if you have any questions, please email us at pdbhelp@ebi.ac.uk, contact us on Facebook at Protein Data Bank, or Twitter at PDBe Europe. Thank you very much.